So now we will look internally at the eye. Internally, it is composed of three layers that are referred to as the tunics. So within the eye, we also have fluids, which are collectively referred to as the humors. And finally, the eye contains a lens, which is going to help to fine-tune the focus of the light that has entered the eyeball. So let's begin by examining the three tunics of the eye. The three tunics are the fibrous tunic, the vascular tunic, and the sensory or the neural tunic. The fibrous tunic composes the externalmost layer of the eyeball, and it's shown here in this diagram in white. The vascular tunic composes the middle layer of the eye, which is shown here in red. And finally, the sensory tunic, shown here in yellow, composes the innermost layer of the eye. So let's begin examining the fibrous tunic, since it's the most external of the tunics, and then from there we will work our way inward. The fibrous tunic consists of two structures. The first is the cornea, and the second is called the sclera. The cornea is a transparent window that's located at the anterior part of the eyeball. And this structure is what allows light to initially enter the eye. The cornea composes only about a fifth of the fibrous tunic. The bulk, that is the remainder of the fibrous tunic, consists of the sclera, which is a tough collagen connective tissue that functions as a site of muscle attachment, but it also provides structure and helps the eyeball keep its rounded shape. Because we'll see in a few moments that the eyeball is actually internally pressurized, and the strength of the sclera is what helps to keep the eyeball shape up against that pressure that's internal. So the cornea is an important structure because it is the major light-bending structure of the eye. So this means that it is the primary structure that focuses light as it strikes and enters the eyeball. It is also the first structure that light strikes as it enters the eyeball because it's the most external. The lens, which we'll examine in a little bit more detail later on, serves to only kind of fine-tune the focus of light before that light strikes the retina and is detected by that retina or perceived by the retina. The cornea itself has no blood vessels on its surface because any blood vessels would interfere with light that is trying to enter the eyeball. The cornea, however, is very richly innervated with sensory receptors, so particularly free nerve endings. So any slight touch or any material such as dust that touches the surface of the cornea will trigger a blinking reflex. The cornea also has some capacity to regenerate if it is damaged. However, if the cornea receives extensive damage, it has to be replaced with corneal transplants from donors. So in much the same way that other organs are donated from organ donors. And this would typically occur immediately after an organ donor has died or passed away. So because the cornea has no blood vessels, this also means that the body's immune system rarely rejects corneal transplants, because the immune system has a very limited ability to reach the cornea and also a very limited ability to reject corneal transplants from other individuals. So next let's examine the vascular tunic, which consists of the iris, the ciliary body, and the choroid. So first let's examine the functions of the choroid. The choroid is a layer of very richly vascularized tissue that's going to function to nourish all of the tunics of the eye. And this is because the choroid is part of the vascular tunic, which makes up the middle tunic of the eye. So it is able to provide nourishment to both the fibrous tunic, which is superficial to it, and the sensory tunic, which is deep to it. So the vascular tunic is in kind of a unique place to be able to nourish all of the other tunics of the eyeball. We see, in particular from this diagram as well, that it is also extremely dark in color, and that's due to the presence of melanin. The melanin pigment functions to absorb scattered light within the eye. So once light passes through the retina, which makes up the sensory tunic, and is also located at the superior part of this diagram, or the top part of this diagram, light passing through the retina gives the retina the ability to form an image, because the retina, as we'll see in a moment, contains sensory receptors called rods and cones, which their function is to detect light. And if light were to bounce off of the choroid, which is located behind the retina, this means that light would once again pass through the retina, and the sensory receptors, that is the rods and the cones, would once again be activated by that light that is passing through the retina one more time. And this could cause the image to be formed to be blurred, or may even cause double vision, because the retina would essentially be detecting the same light twice, as it first strikes the retina and then bounces off the choroid and strikes the retina again. So the melanin that's located in this layer helps to absorb light after it has passed once through the retina to make sure that that light does not bounce off or scatter from the choroid. So a structure that is seen in nocturnal animals is a structure within the choroid that is called the tapetum lucidum. This is a Latin word that means the bright tapestry. It is usually very bright blue or green iridescent in color. We don't see this in the human eye, so humans do not have this. But animals that are active at night, like deer, cats, dogs, and cows, do have it. And this structure helps to improve vision in dim light as well as dark habitats. So here's a photograph that you can very easily see that there are 4 cats in this photograph because of the 4 pairs of eyes. The camera flash has bounced off of the tapetum lucidum of the cat's eyes, making the cat's eyes appear to flash bright blue or bright green for a moment. So let's continue our examination of the vascular tunic by turning our attention to the ciliary body. The ciliary body is another main component of the vascular tunic. It is primarily composed of bundles of smooth muscle, and those bundles of smooth muscle are attached to the lens by small ligaments called the suspensory ligaments. The smooth muscle is controlled by the autonomic nervous system. The muscle can be stimulated to contract, which actually causes the suspensory ligaments to go slack, and in doing so, that will cause the lens to become more rounded and bulge. So if the muscles relax, the tension on the lens increases and the lens becomes flatter as a result of that. So as the lens changes shape, the ability of the lens to focus light on the retina will also change. And we're going to see how this works a little bit later within this segment. So the next structure that we're going to examine is called the iris. And the iris is the anteriormost structure of the vascular tunic. Its name means rainbow in Greek. The anterior part of the iris, that is the part of the iris that we can see through the cornea, is pigmented. So when we discuss the color of someone's eye, we are typically referring to the color of the irises of their eyes. The majority of the iris is actually composed of smooth muscle. A small opening at the center of the iris, which is called the pupil, controls the amount of light that enters the eyeball. So in dim light, the pupil will dilate, and in bright light, the pupil will constrict. And what this does is this allows enough light to enter the eye so that an image can be formed, but it does not allow so much light into the eye that the image gets washed out or the retina becomes damaged. So extremely bright lights, or even looking directly at very bright objects like the sun, can actually permanently damage the retina, and so it's important that enough light gets let into the eye so that an image can be formed, but not so much light that the retina could potentially become damaged. The variety of colors that the iris can take on is quite variable, and there are many genetic factors, that is, many factors within our DNA that contribute to an individual's iris color. Brown, blue, and green are fairly common, but the iris can also be amber colored, or even red or purple colors are possible as well. Melanin is the pigment that contributes to eye color, and melanin also helps to protect the deeper structures of the eye from damage by sunlight. So, however, the wide variety and the other roles that eye color play are not yet known. So the final and the deepest tunic of the eye is a structure called the sensory or the neural tunic. It consists of a single structure referred to as the retina. And the retina is where the eye's sensory receptors are located. And collectively, these receptors are called photoreceptors because the term photoreceptor implies that they're sensitive to light. So here we see that the retina is situated at the deepest part of the eye. We can see that there's blood vessels that sit on its outer surface. Immediately behind it is the choroid, which helps to nourish the retina as well as the sclera. And we're going to see that there's a very specific layer of the retina that contains the photoreceptors, while the other layers of the retina contain nervous system cells as well as the axons of sensory neurons that are going to communicate the images and the information that the photoreceptors are gathering to the brain to be interpreted. So the photoreceptors are arranged towards the back of the retina. So light striking the retina would first pass through a couple of layers of cells. The first is a layer of cells called the ganglion cells, and then another is a layer of cells called the bipolar cells. And so that light would pass through those before making contact with the photoreceptors, and this light making contact with the photoreceptors is what will activate them. We see that there are two kinds of photoreceptors. There are the rods, which are shown here in purple, and then there are the cones, which are shown here in yellow. We'll examine the functions of these two photoreceptors in just a moment. So any light that passes by the photoreceptors without striking them and without activating them would then be absorbed by the melanin that's located in the pigment layer of the retina. This light could also be absorbed by the melanin that's located in the choroid as well. So in total, there are about 250 million photoreceptors, that is both rods and cones, in the retina. The photoreceptors that are called cones function in color vision. So certain cones are sensitive to specific wavelengths of light. So for instance, there are red, green, and blue cones that are sensitive to red, green, and blue light, respectively. So visible light particles travel in waves, and so therefore we measure the different types of visible light as wavelength. Blue cones are most sensitive to light, which is shown here as absorbance, in the 420 nanometer range. And the 420 nanometer range of wavelength corresponds to violet and blue light. Green cones are most sensitive to light in the 530 nanometer range, which corresponds to green light mostly. And red cones are most sensitive to light in the 560 to 570 nanometer range, which corresponds to kind of the yellowish end of the spectrum, but then you can also see that the cones do have, the red cones do have some sensitivity higher up in the spectrum, the wavelength of visible light here. So they are able to detect yellows, oranges, as well as reds. Next we have rods, which are the second type of photoreceptor, and rods function best in dim light. They are unable to sense the color of light, and because of this, the visual images that are produced by rods are gray in color. That is, they take on a grayscale appearance, so shades of dark gray or shades of light gray. The distribution of rods and cones is not even across the retina, and we'll see this illustrated in kind of a map of the retina on the next slide here. In this slide here, I wanted to examine two structures. So at the posterior aspect of the eyeball, that is at the posterior part of the retina, there's a small oval spot that is called the macula lutea, and in the center of the macula lutea is a tiny pit that's called the fovea centralis. And the fovea centralis, or quite simply the fovea, contains a very high concentration of cones. And in the fovea, because there's such a high concentration of cones, visual acuity is greatest for images that fall on the fovea. So that is, when you're trying to examine something in high detail, you position your eyes so that the light coming from that object is focused on the fovea centralis. And so that's what gives us kind of our greatest acuity and definition for shapes and colors and things like that in our vision. The remainder of the retina that is in the periphery of the retina from the fovea centralis, we see that the retina contains a more higher concentration of rods in those places rather than cones. So in this diagram on the left, we see that the receptor density is mapped on the vertical axis, with higher numbers corresponding to greater receptor density, that is, greater numbers of either rods or cones. Here I'm not concerned with you recalling the exact number of rods and cones. I just wanted to illustrate the fact that rod and cone density does change as you move around the retina. We also see that distance from on this graph on the left here, that distance from the fovea is plotted on the horizontal axis. So we see at the fovea is the highest density of the cones, which is shown there by the vertical red line there. And as one moves away from the fovea towards the periphery of the retina, the rods become more numerous. And so what that means for our vision is that objects that we want to see in high detail, so with color, but also high clarity and high definition, we position our eyes so that our eyes focus the light coming from that object, and the light coming from that object is focused onto the fovea centralis of the retina. Because rods are responsible for vision in dim light environments, that means that our nighttime and low light vision is best in our peripheral vision, because we see that rod density is highest in the peripheral parts of the retina. Cones are unable to operate correctly in low or dim light environments, so we cannot see in the dark with these types of photoreceptors. So looking at an object directly in the dark is actually not beneficial because we effectively have a blind spot in the center of our vision when we're in low or dim light environments, because the cones are not going to be active in low or dim light environments. So they would be unable to detect the objects in our visual field if the environmental light is low or very dim. So as we saw previously, the retina receives blood supply from a couple of sources. The first we saw are blood vessels that are in the choroid, and the second are the extensive network of blood vessels that sit on the surface of the retina. These small blood vessels that sit on the surface of the retina are the branches of the central artery and the central vein of the retina. Next, the structure that we'll turn our attention to is called the optic disc, and this is a structure where the neurons that are in the retina form a bundle and exit the eye as the optic nerve. There are no photoreceptors at this spot, so functionally this is a blind spot. So any light that is focused on this part of the eye will not be detected, and so no image can be formed from those. However, we do not notice these blind spots or these gaps in our vision because our brain uses a very sophisticated process to fill in the missing visual information from that part of our vision. So you can very easily test for the location of this blind spot in your visual field. So in this demonstration here, use your left hand to cover your left eye. And then once you've done that, what you should do is fix your right eye on the red circle to the left. So keep your left eye covered and your right eye looking at the red circle. And while you do that, slowly move your head closer to or further from the screen. So don't move it up or down, but just move it back and forth, roughly at kind of the same level. Put your head at the same level as the slide here. And what you'll notice as you move your head back and forth very slowly is that at some point the red X that is off to the right will disappear from your peripheral vision. And so when that red X disappears, it means that that red X is in your blind spot. So the inside of the eye is divided into two cavities. So we can now take a look at these cavities since we have finished exploring the tunics of the eye. The anterior cavity is a cavity that's located anterior to the lens, and the anterior cavity is divided into two chambers, the anterior chamber and the posterior chamber. The anterior chamber of the anterior cavity is located between the cornea and the iris. Meanwhile, the posterior chamber of the anterior cavity is located between the iris and the lens. The anterior cavity is filled with a fluid called the aqueous humor, which is a watery liquid that is derived from the blood. The aqueous humor pressurizes the eye, and that pressure helps to give the eyeball its shape. The aqueous humor, because it comes from the blood, it also functions to nourish the lens and the cornea, because neither the lens or the cornea have blood vessels on their surface. The posterior cavity is filled with a crystal clear gel-like fluid that is called the vitreous humor. The functions of the vitreous humor are to hold the retina against the choroid. That is, they physically press the retina against the choroid and hold it there. And the vitreous humor also functions to transmit light from the lens to the retina. So here we'll examine how the aqueous humor is produced. The aqueous humor comes from blood vessels that are found in the ciliary bodies. It flows from the posterior chamber to the anterior chamber. And in the anterior chamber, it is reabsorbed by small veins called the scleral venous sinus. And these scleral venous sinus are located at the junction between the cornea and the sclera. So if the aqueous humor is not properly drained, that means that excess aqueous humor can accumulate in the eye and increases the pressure inside of the eyeball. And this condition is referred to as glaucoma. The elevated pressures in the eye can also put pressure on the vitreous humor, and that vitreous humor will then press harder on the retina at the back of the eye, and it could cause the retina to slowly die. The aqueous humor functions, as we've mentioned before, to maintain pressure within the eye, and also supports the eyeball internally. Its other functions include supplying nutrients as well as oxygen to the lens and to the cornea, because both of those structures lack blood vessels. The vitreous humor that is in the posterior cavity is formed during fetal development, so it is not replaced like the aqueous humor is. So normally, as shown on the left, aqueous humor will drain from the eyeball. So it will be produced, it will circulate, and then it will drain through those scleral venous sinuses. However, blockages, which are shown on the right, that are going to prevent proper drainage of the aqueous humor, will cause conditions such as glaucoma, where the retina and the optic nerve are slowly crushed because of the excess pressure inside of the eye. And when they are crushed, that will cause them to slowly die due to the increased pressure that is accumulating within the eyeball. So next, let's turn our attention to the lens. And the lens is a flexible structure that is composed of fibrous proteins as well as crystal-like minerals. It is supported by the suspensory ligaments of the ciliary body that are located just behind the iris. The smooth muscles that are in the ciliary bodies can contract and relax, and therefore change the amount of tension that those suspensory ligaments put on the lens. The change in tension will change the shape of the lens, and that gives the lens the ability to more precisely focus light onto the retina. So just like the cornea, the lens is avascular, so that is, it has no blood vessels, because any blood vessels on its surface would interfere with the transmission of light through the lens. And so this is why it's important for the aqueous humor to fulfill its function of nourishing the lens tissue. New lens fibers and minerals are added continually throughout our life, so that means that as we age, the lens is going to get thicker and much tougher and become a lot less flexible. And so therefore that's going to interfere with its ability to fine-tune the focus of light on the retina. A specific condition that can affect lenses is called cataracts, and cataracts is a clouding of the lens, which usually results from age-related thickening of the lens, or it could also be a secondary consequence of untreated diabetes mellitus, because that can cause the lens crystals that are deposited to be cloudy. So the pathway of light that light takes as it enters the eye is as follows. So first it's going to strike and pass through the cornea. It's the first structure that it's going to come in contact with. And then it will travel through the aqueous humor, through the pupil, and then after that it will strike the lens. and then after striking the lens, it will proceed through the vitreous humor before finally landing on the retina, where it will be detected by the photoreceptors. So light that enters the eye is bent and focused by two structures, as we mentioned. The first is the cornea, and the second is the lens. Because of the curvature of the cornea, and because it is also the structure that light will strike first when it enters the eye, the cornea is the major light bending or light focusing structure of the eye. And we see that the lens only serves to fine-tune the focus of light that's falling on the retina. So lenses, including the cornea and the actual lens of the human eye, as well as corrective lenses, are structures that bend light, right? So the point at which all of the light rays from an object coming together, the point at which all of those light rays that are being emitted from an object or bouncing off of an object that are focused by the lens at a specific point is called the focal point of that lens. So if an object is to appear clear, that is, if the light that is focused and lands on our retina is to appear clear and not blurry, that means that the focal point of the light rays that are being bent by the cornea and the lenses the focal point of those light rays has to land on the retina. If the light lands on the retina at a point other than the focal point, then the image that's going to be formed is going to be blurry. So now let's briefly examine how the eye focuses on distant objects as well as nearby objects. This is controlled by the two divisions of the autonomic nervous system. So when the sympathetic division of the autonomic nervous system causes the smooth muscle in the ciliary body to relax, this increases the tension on the suspensory ligaments, which here are referred to as the ciliary zonules. The increased tension on the suspensory ligaments cause the lenses to be pulled, and in doing so, the lens becomes thinner and flatter, which helps our eye to be able to focus on more distant objects. So we see that when trying to focus the focal point of the light on the retina from a distant object, the lens will actually become thinner and flatter as a result of that. So on the other hand, when the parasympathetic control causes the ciliary muscle to contract, this is going to cause the suspensory ligaments to go slack. Less tension on the suspensory ligaments will also cause the lens to become more rounded and bulge. So a more rounded lens helps to focus on nearby objects. So we see that these changes in lens shape will help us with vision of different objects at different distances. So two conditions that can result from the focal point of the light not landing on the retina are called myopia and hyperopia. The first we'll examine real briefly here is myopia. Myopia refers to nearsightedness, which is a condition in which nearby objects appear clear, but distant objects appear blurry. And this is a result of the eyeball being too long, because light from distant objects is focused on in front of the retina rather than on the retina. So it is the focal point of the light that's coming through our cornea and through our lenses is too far in front of the retina. The focal point is not on the retina. So this means that light that strikes the retina is out of focus, which causes our view of distant objects to appear blurry. So notice that in this example, the corrective lenses simply move the focal point further back so that light, by the time it reaches the retina, is at its focal point, rather than being in front of the retina. And last but not least, we have hyperopia, which refers to farsightedness. This is a condition in which nearby objects appear blurry, but distant objects are focused correctly on the retina. This is essentially the opposite problem of myopia. So with hyperopia, the eyeball is too short, so that means that light from nearby objects is focused behind the retina. Light from distant objects is going to be focused correctly on the retina. And here, corrective lenses are going to fix the problem by moving the focal point of light forward so that that focal point lands correctly on the retina. So both myopia and hyperopia can be corrected by corrective lenses or by a procedure called LASIK, which is a procedure that uses lasers to reshape the surface of the cornea so that the focal points of light from nearby or distant objects will land properly on the retina.